All right. Hello. Is everybody ready? Yeah. All right. Okay. So I'm Seth, and I'm doing research this year, and I'm here to share with you guys what I kind of learned, and then my process, and then so first, before I get started, I wanted to thank my mentor, Dr. Rebecca Prescott, for helping me through everything, and even though she's like 11 hours ahead, and always she's in Australia, and she's always there when I text her or when I like email her. She's always there, and yeah. So I just want to give her a quick thank you. So I'm doing quorum sensing in cyanobacteria. So you know that cells communicate with each other, single-celled organisms, and well, yeah, they do. And so what they do is they release and they detect chemicals in the environment, so it helps them coordinate their behavior. So quorum sensing was first discovered in 2005 in the Hawaiian bobtail squid. It allows the cells in the squid to glow when they reach a certain density. So on top of this, we also see quorum sensing in the biofilms in your teeth and the plaque in diseases such as strep throat. And quorum sensing, so I guess to understand why we call it quorum sensing is, if you think about a quorum is a minimum amount of people for a certain action. And so that's why it's called quorum sensing, because it's a behavior based off of the density of the chemicals in a given space. So let's get started a little bit. So I'm talking about the LuxR gene today, or the system today. So the LuxI gene, it is the protein, it releases the protein that releases the chemical that allows cells to talk to each other. So you can think of it as the mouth of the cell. And then the LuxR gene, it produces the receptor which allows the cells to listen and detect changes in the environment. So you can think about it as, I guess, the ear of the gene, yeah. So I guess to look at this more closely, I picked cyanobacteria. So the reason why I picked cyanobacteria is because they're one of the oldest known organisms, and it's also a huge sample group for me to choose from. But cyanobacteria, fossils date back up to 3.8 billion years, and no one's really looked at quorum sensing in cyanobacteria yet. So I thought that I would try to take a different approach and try and find the genes for quorum sensing in cyanobacteria. So I guess the general question, sorry, I'm getting kind of redundant. Do cyanobacteria have LuxI, LuxR sensing systems, and if so, what purpose do they serve? So this is kind of a broad question, so I'm going to break it down into a couple of different parts. So first off, are LuxI and LuxR both present in cyanobacteria? And then are LuxI and LuxR present in all cyanobacteria? And how many copies of the gene are there per organism? And then how are the genes evolving and changing, and how is quorum sensing in Vibrio fischeri, which we found in the Hawaiian bobtail squid, different than quorum sensing in cyanobacteria? So before I go about answering these questions, I'm going to tell you about how I answered these questions, at least. So first, I downloaded these sequences downloaded whole genome sequences of cyanobacteria from GenBank. So GenBank is this big, huge public database that has all these genome sequences, and it's really helpful, and it really allowed me to get a really good, nice, big sample size. And then from there, I ran Rocker. So what Rocker is it's a software that was developed to search for certain genes in whole genome sequences from GenBank. And so from there, I had a script, and so that script pulled down it took the information, and then it pulled down the genomes, or it pulled down the genes out of the whole genomes. And so that just was so I didn't have to do it all by hand. And then from there, I aligned the sequences with confirmed LuxRs from Signal. So what that means is Signal is a database of quorum sensing genes that have been tested and vetted and confirmed. And then from there, after I aligned them, I constructed phylogenetic trees, and I'll show you guys those in a little bit. So now we can get to what I did actually find out. So to answer the first question, do cyanobacteria have quorum sensing genes? Well, first off, they don't have the LuxI gene. I didn't find it in my sample group of 135, so it is possible that they do have it, but in my sample group there wasn't any. And in 105 LuxR genes were found in 135 cyanobacteria sequences. However, these LuxR genes were only found in about 45 different cyanobacteria sequences, as some organisms had up to 10 copies of the LuxR gene. And so I guess what the absence of the LuxI says is that these LuxRs are LuxR solos. And so it allows them to, I guess, eavesdrop on what's going on around them. Whereas if they had the extra, or the, sorry, if they had the Lux Eye, then they would also be able to talk. But in this case, they're only able to listen. All right, so here are two chromatograms that I have. And so the top graph came from one organism. And so if you look at the yellow part, the yellow and red part at the bottom of the top graph, the higher the peak, it means that the sequences were more similar, and then the lower the trough, it means that the sequences were very different. So since this was all within one organism, it kind of shows that there is variation even within the organism, within organisms themselves. And then in the second one, you can see, or in the bottom graph, you can see kind of a similar evolutionary trend as the one above it. And then I'll go a bit more into depth about that now. So this is the first phylogenetic tree. So it has 7 sequences, and these all came from one organism. So you might be wondering, why do they all have, or why are they different? Or if they all came from the same organism, why is there so much variation? So it's a good question, but if you think about it, why would an organism have the same exact copy of a gene multiple times? It's useless. So what actually happens is that these organisms have multiple copies of the LuxR gene that do different things, and so we see that in this kind of variation here. So, like I was talking about earlier, if you look at the peak and the trough, you see kind of housekeeping, I guess you would call them, portions of the gene. And so on those higher peaks, it means that that portion of the gene is necessary for it to be a LuxR, for it to do its job. And then for the lower parts, those are parts that can vary more. There's more wiggle room for them to be able to change and, I guess, evolve with the organism. And so, oh, I'm sorry, I had an animation, but I guess it, so, sorry, it threw me off a little bit. So you can see the same thing here, and if you look at the evolutionary trend again, it goes up and then down. So these higher peaks, it's parts where it's conserved. And so what this kind of tells us is that these low areas of variation, it allows for the organism to, I guess, change based on the environment that it's residing in. And so I have some conclusions. And so I guess to conclude, there is quorum sensing in cyanobacteria. And this shows that quorum sensing could be necessary for survival, and it's significant. It was significant before, and it's significant now. And so before now, we didn't know whether or not cyanobacteria participated in quorum sensing or even had the genes to do it. But as we see that quorum sensing is present in a wide range of cyanobacteria, it likely evolved once in a common ancestor, and then remained in the gene pool and then kept getting passed down. All right, so I guess before I get to thank yous, what I learned was the research is hard. It's a lot of work, and it really does take a lot to get through, and it doesn't sound like a lot. Like when you're researching for a paper or when you're researching to do a project, it's really completely different from doing independent research. You can't Google what you're going to do next because this is all brand new. You're paving the way. And although it may be difficult, it's really rewarding. It feels good when you're done. You may have lots of late nights and lots of sleepless nights, actually. But but in the end, when you have your poster printed out, or when you have a slide, or a slide presentation, it really is worth it. And if any of you have doubts about taking research next year because of the amount of work, I really implore you to take the class, because if it does come out to become too much work for you, then I guess cut back a little bit and then you can drop it at the beginning of the year. But if you have any doubts about it, you should just try it out. And so I guess as a nice segue, I would like to thank everybody that put up with my grouchiness for being up so late working on this. And so I would like to thank my mentor, Dr. Prescott, and also my research teacher, Dr. Chen, for helping me and then being there when I emailed, frantically not knowing what to do. And then also my mom and my dad for all their love and support through everything, and then congratulating me when I finally got something done. And then lastly, I would like to thank my research class, who has really been an important part in this process, and they help me. I guess they motivate me, they keep me going, because I know I'm not alone. I know I'm not the only one that's struggling. There's lots of other people struggling too, and it feels good to, I guess, know that they're all there for you. And so, yeah, that's how. Thank you.